Welcome to the all viewers and well-wishers of the world. Purpose of my lecture is to learn the knowledge from the word and return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot. Now it's time to return a lot to the word from where I have got the knowledge. Topic of today is bacterial genetics. Bacterial genetics means exchange of the genetic information in the bacteria from one bacteria to the another, and the objective of the lecture is nature of the different transposable genetic material or elements, mechanism of the gene transfer in the bacteria, and the significance of the transfer of genetic material in bacteria. Genetic material consists of four things. One is chromosomal DNA, the other is plasmids, the third is transposons and transposable genetic material like insertion factor from one bacteria to another, and then genes which can be transferred from one bacteria to another. So the first chromosomal DNA, it is a single large circular chromosome haploid attached to the cytoplasmic membrane. The septal mesosome replicate by the simple fission instead of the mitosis as in the human cells. It has got 2000 genes. A human DNA has about 100,000 genes. Advantage of the 1N DNA over the 2N DNA is that it is more efficient and it grows more quickly. And chromosome DNA has no histone proteins, no introns as in eukaryotic DNA, but polyamines and the magnesium ions which work like eukaryotic histones here in bacteria that can be realized with fusion DNA staining as oval or the elongated body in the bacteria. Unfolded nuclear DNA is about 1 millimeter long. So here one can see the chromosomal DNA. The bacteria do have this chromosomal DNA, and here again the bacteria have chromosome, and it also has the plasmid, and here it is a plasmid. Here one can see the original staining of the bacteria showing the chromosome by the stain which is used to just visualize this. Now the number two is plasmids. These are extra chromosomal double-stranded circular or linear DNA molecules can integrate or merge with the bacterial chromosome. It is capable of self-replicating independently of the bacterial chromosome. It occurs in both gram-positive and gram-negative bacteria. It carries genes for the following functions and the structures. These are antibiotic resistance or R factor, resistance to the heavy metals such as mercury, which is present in the antiseptic, so the bacteria cannot be killed by these antiseptic. Here one can see the plasmid. These are rounded mostly in shape and resistant to the ultraviolet light, pili, fimbriae, which mediate adherence of the bacteria to the epithelial cells, that is mediated by the plasmids. Exotoxin, including several enterotoxin, again under the influence of this plasmid. Episome is a plasmid that can integrate into the chromosome of the bacteria. Different types of the plasmid can exist in one cell. These are transmissible plasmids or mobilizable plasmid. Simply the name indicates. It can be transferred from cell to cell by the conjugation or the sex pilus, and the other is non-transmissible that cannot be transferred, so do not contain transfer genes. And here one can see the plasmids, the mobile plasmid, and here it is that sex pilus through which this can just give this part of this plasmid to this cell which is having the no plasmid. So this is the donor and this is just transmitting through the F or sex pilus to this recipient bacteria, and here now the relaxosome is being and the transferosome are being transferred to this recipient cell, and recipient cell, after receiving this in the form of this plasmid, that it becomes the new donor, and here it can see that it can work like this to transfer the genes from this bacteria to the other recipient bacteria. The transposons, these are the small mobile DNA pieces, move between the chromosomal DNA and the plasmid DNA. The process is called transposition from one position to the another position of the chromosomal DNA and the plasmid DNA. These are nicknamed jumping gene due to their ability to move. They cause mutation in gene, so insert or alter expression of the nearby genes. They are not capable of self-replication. They carry genes for the antibiotic resistance, toxins, and the certain enzymes being produced by the bacteria. Several transposons can be located in the DNA. Here one can see transposon. These are present on this DNA, and that can be transferred from this part to this part. Similarly, this part of the chromosome, that the transposon, the piece of the chromosome that can be transferred to the other piece of the chromosome. Similarly, this can be transferred to the plasmid also, that is also the extra chromosomal part of the bacteria. So this transposon, that are the pieces of the genetic material transferred from one part, either of the same part of the chromosome or from the chromosome to the plasmid. The number 4 is the gene. The unit of heredity, a segment of the DNA that carries in it the nucleotide sequence. It is the information for a specific biochemical or physiological property. Bacteria has 2,000 genes. Human DNA has 100,000 genes. Usually, gene transfer is between members of the same bacterial species. And here one can see the transposon is just having one gene, and that is just gene is going to be cut from this transposon, and it is going to be pasted to the chromosomal DNA, and this cut and paste of the gene, that is the gene transfer from this transposon to the chromosomal DNA. Similarly, one can see the DNA, the transposon that is being transferred from here to this DNA to this DNA, and that is disrupted gene, and that is being transferred from this DNA to this DNA. So that is the gene transfer. The nice thoughts are that kill the tension before tension kills you. Reach your goal before the goal is away from you and the goal is going to kick you. Live life before the life leaves you, and life does not wait you. You have to do something extraordinary to get something. Now, exchange of the genetic information. It is the natural mutation in the DNA of the cell, or it is the acquired, that is the acquisition of the additional DNA from an external source, and that is by 3 processes which I am going to tell you in detail, that is transformation, transcription, and conjugation. So, first, the natural mutation in the bacteria. Genetic determinants of an organism constitutes its genotype, that tells the genetic makeup of any bacteria, and the observable characteristic like flagella, fimbriae, and the other that constitute its phenotype. Mutations are the change in the genome or the genotype, that is chromosomal DNA, plasmid, genes, and other things, but not the phenotypical changes. Here, genes can be transferred to the other bacteria. A mutation arising in one cell can be passed on to the other cells during the division and to have the same characteristic as the mother was having, that is going to be transmitted to the daughter. Organisms without any change are called wild type, and with mutation are called mutants. So mutants are those which get some information from the mother cell. Here one can see the exposure to bacteria occurs and infection occurs and the bacteria spread, and ultimately when the drug is fired during treatment, that bacteria do die. They are not capable of replicating. But for the survival, drug resistant bacteria, they do take some gene from the other bacteria. As one can see, the red color of the plasmid, it is having some resistant genes, and these bacteria multiply and these become having larger number with that mutant genes. And here one can see when the antibiotic is given, they are able to resist and the bacteria continue to spread and the person remains sick, but the bacteria do continue to spread, divide because of this gene which is present there, and the antibiotic cannot kill these bacteria. And here again one can see the different kinds of the bugs and the superbugs which are present in the bacteria to resist their killing by the antibiotic. Here again non-resistant bacteria exist and the bacteria multiply by the billions, and here some mutation make the bacterium drug resistance and the remaining are able to be killed by the antibiotic, and these two which have survived, they are having the drug resistance and the bacteria multiply, and here one can see they can survive and thrive even with the antibiotic. Natural mutation in bacteria, that is selective and differential media, are helpful for isolating bacterial mutants from the cultures. Mutation arise in the bacterial populations. These can be spontaneous, that can be checked by the flocculation test, and it can be induced by some chemicals or the physical treatment to the bacteria to cause the natural mutation in bacteria. Mutation can be harmful, lethal, helpful, or it can be silent. Processes involved in the mutation are nucleotide substitution, microdeletion, microinsertions, and rearrangements. Acquired exchange of the genetic information is the other way by which the bacteria do acquire the genetic information. Now, bacteria have three ways of exchanging genetic material or DNA between the bacteria, as I have already told you. Now here it is the transformation. The recipient bacteria takes up extracellular donor naked DNA. Transduction. The donor DNA packaged in a bacteriophage infects the recipient bacteria and transfer the DNA through the bacteriophage to the recipient bacterium. Conjugation. It is a donor bacterium transfer DNA to the recipient by the pilus, and mating, it is called the bacterial sex. I will tell you in detail the every part. First, the transformation. It is the gene transfer resulting from the uptake of the DNA from a donor. Pieces of the DNA released from the donor bacteria when the bacteria dies or lysed, these are lying in the environment, and these are being taken up by the competent recipient bacteria directly from the extracellular environment. It can occur with DNA plasmid. Many factors like temperature of the salt solution affect this transformation process. Transformation was first discovered in Streptococcus pneumoniae bacteria. Here one can see the transformation. The DNA fragments of the dead or the lysed bacteria, the red color are lying here, and the competent bacteria having the chromosome, it takes that DNA fragment which are lying naked here, and here it is taking up the DNA, the fragment, and integration by the non-reciprocal recombination. And here one can see this part of the DNA is trying to merge with this chromosome, or it is not going to merge with this chromosome. So these two conditions, one is called stable transformation, as one can see there is a mergence, there is the merging of this piece of the DNA fragment with this chromosome, and here that is being broken up, not taken up by the chromosome. So this is called unsuccessful transformation. Here again one can see transformation with a plasmid. The DNA plasmid, as you can see, the circular, that is just going to be taken up by the bacteria, and when the uptake is complete, that is going to cause the stable transformation, as the plasmid is lying successfully in the bacterium with the chromosome, and it can merge or not merge later on. So transformation occur in bacteria like Haemophilus, Neisseria, Bacillus, and Staphylococcus. Transformation occur in the nature and lead to the increased virulence, and it is widely used in recombinant DNA technology. And transformation is stable if DNA fragment gets merged with the recipient chromosome, as I have already shown you in the previous slide. Unsuccessful transformation when DNA fragment do not merge with the recipient chromosome. Here again one can see the bacteria transformation with plasmid DNA. You can see the plasmid is going to merge with the chromosomal DNA to make the stable transformation, and here one can see the DNA pieces, they just do join the chromosome to make the stable, and when they are broken up into the pieces and not able to join the chromosome, that is called unsuccessful transformation. So steps in transformation are: number one, uptake of the DNA; number two, it differs in gram positive and gram negative bacteria. Then in gram positive bacteria, DNA is taken up as a single stranded molecule, and complementary strand is made in recipient. And in contrast, the gram negative bacteria take up double stranded DNA, and legitimate homologous or general recombination occur in transformation, and after taking donor DNA, recombination occurs between chromosome and the DNA. And this recombination requires homology between the donor DNA and the recipient chromosome. If they will be having the different nature, they will not be able to make successful and stable transformation. Now, there are certain factors which affect the transformation. These are which help DNA taken up and merging into the recipient chromosome. These are the certain factors, and it depends upon the size of the DNA and the state of the DNA. Then double stranded DNA of at least 5 into 10 power 5 Dalton works best for this mergence and transformation. They are sensitive to the nuclease in the environment, and they are just not going to be killed by these. And then competence of the recipient, whether they are competent enough to take these this transformation or to take the pieces of the chromosome and other, can be spontaneous or induced, or bacteria produce a specific protein called competence factor, which is responsible to take these pieces of the DNA or the plasmid. These are the bacteria which are having this specific protein. These are Bacillus, Haemophilus, Neisseria, and Streptococcus. And it can also be produced by the physical treatment with the calcium chloride to make these cells competent, which were previously not competent. Here one can see the non-competent bacterium through the environmental signals, they become competent. Now they are competent enough to take the free DNA which is found in the environment, and that is this bacteria with competent DNA is able to have free DNA, and that has formed the transformed bacteria, and one can appreciate this blue piece of the free DNA that has merged with chromosome to form transformed bacteria. Here again, transformation factors are certain treatment with the different bacteria like salt solution and calcium chloride, and other treatment with the different temperature using, and that is just going to make the bacteria competent enough to take the different DNA pieces, and these are just acting as the transformation factor, and the bacteria undergo the transformation.